Histology of the Thymus gland. Hi, here's another lesson on the histology of the lymphatic system. In our previous videos, we covered two of the secondary lymphoid organs, namely the lymph node and the spleen. In this video, we have one of the primary lymphoid organs called the thymus gland. The thymus is an extraordinary gland nestled within the chest cavity in the upper anterior mediastinum, just behind the sternum and between the lungs. It plays a crucial role in the development and maturation of T cells, a vital component of the immune system. During early childhood, the thymus is at its largest size and gradually decreases in size as one reaches adulthood. The thymus has a unique structure that sets it apart from the other organs. It is composed of two distinct lobes, each further divided into smaller lobules. It is encapsulated by a connective tissue capsule. Histologically, the thymus consists of two main compartments, the cortex and the medulla. Let's discuss the features of the cortex. The cortex is located toward the outer region of the lobules and is densely packed with developing T cells known as thymocytes, which are the principal cell type within the cortex. They originate from precursor cells that migrate from the bone marrow to undergo a series of steps of maturation within the cortex, transitioning from immature cells to mature T cells. The cortex acts as a checkpoint for thymocyte development, allowing for the selection and survival of T cells that display functional antigen receptors. Thymocytes that complete these developmental checkpoints progress towards the medulla for further maturation. The other cells we find in the cortex are the thymic epithelial cells. They form a network throughout the cortex, interacting closely with thymocytes, and play a critical role in the development and education of T cells. And now we move on to discuss the features of the medulla. The medulla is located toward the central part of the thymic lobules, surrounded by the cortex. It appears less densely packed than the cortex and often has a lighter staining appearance under the microscope. The medulla contains a mixture of different cell types, including more mature T cells, specialized epithelial cells, and other stromal cells. T cells that have successfully undergone positive and negative selection in the cortex continue to mature. These T cells, now more mature and functional, make up a significant portion of the medulla. They display a diverse T cell receptor repertoire and are capable of recognizing foreign antigens. Within the medulla, Hassel's corpuscles, also known as thymic corpuscles, or medullary thymic epithelial cell nests, can be observed. These structures appear as concentrically arranged walls of keratinized epithelial cells. Let's talk about the positive and negative selection of the T cells. Within the cortex, thymocytes undergo positive selection, a process that ensures the generation of T cells that is capable of recognizing foreign antigens, while also maintaining self-tolerance. Negative selection occurs within the cortex. Thymocytes that strongly react to self-antigens are eliminated through apoptosis. This removes T cells that could potentially cause autoimmunity. In the medulla, T cells undergo further negative selection to remove any remaining autoreactive or self-reactive T cells that may have escaped elimination in the cortex. Moving on now, let's discuss the clinical anatomy relevant to all the lymphoid organs we discussed. Lymphadenopathy refers to the abnormal enlargement of lymph nodes. Clinically, it can manifest as swollen and palpable lymph nodes, often indicating an immune response to infection, inflammation, or malignancy. Lymphedema is a condition characterized by the accumulation of lymphatic fluid due to impaired lymphatic drainage. It typically leads to swelling, usually in the extremities. Splenomegaly refers to an enlarged spleen caused by various conditions, including infections such as mononucleosis, liver disease such as cirrhosis, hematological disorders such as leukemia or lymphoma, or autoimmune disorders. Sentinel lymph node biopsy is a surgical procedure performed in certain types of cancer, like breast cancer, to determine whether cancer cells have spread to the nearby lymph nodes. The procedure involves identifying and removing the sentinel lymph node, which is the first lymph node that cancer is likely to spread to from the primary tumor. Thymoma is a rare tumor that originates from the cells of the thymus. It can be benign or malignant. Pop quiz. That concludes our session on the histology of the lymph nodes, the spleen, and the thymus. We hope you had fun learning with us.